Hi, everyone. Welcome. Today we have really a pleasure to listen to Yoshiko Kojima, who I've known for a couple of decades. I came across her work when she was a master's student in Japan in Tsukuba University, and she was working with monkeys, and she made this amazing discovery, which later was named as spontaneous recovery of memory. And that was really instrumental in helping us understand time courses of learning in the brain, particularly the cerebellum. So she finished her Ph.D. from University of Tsukuba with Professor Yoshida. Then America was lucky enough that she came to the United States. She came to the University of Washington in the laboratory of Albert Fuchs, and she began learning about neurophysiology of the cerebellum, and has been really a prominent and one of the most important contributors to understanding the role of the cerebellum in control of saccadic eye movements. It's great to have you, Yoshiko.
Thank you. Thank you very much. So, good morning, everyone. We have been studying saccade adaptation, and especially like visually guided saccade adaptation and memory guided saccade adaptation. And recently we started covert saccade learning. So I think they are all saccade learning or saccade adaptation, but they are quite different. So for example, learning type, the visually guided saccade adaptation is a gain control of like sensory signal to motor signal transformation, like in this case, visual signal to the saccade signal. But memory guided saccade adaptation, this is also gain control, but I think this is the spatial memory signal to the motor command, saccade command signal transformations gain control. But anyway, both adaptations are for securing the movement accuracy. Covert saccade is probably acquisition, we don't really know yet, but this is a covert saccade is a compensatory saccade after vestibular dysfunction, which relieve vestibular symptom. So in the last time in this seminar, I talked about visually guided saccade adaptation. So today I am going to talk about this memory guided saccade adaptation. Maybe this next time. All right, so first I'm going to show you the 5 different types of saccade. There are many more, but just 5 types here. So targeting saccade or visually guided saccade, express saccade, delayed saccade, and memory guided saccade, and scanning saccade. So targeting saccade is the most basic saccade that monkey or human is fixating on the target and the target step, and monkey makes saccade to the target. And express saccade can be induced by so-called gap paradigm, and which is while monkey is fixating on the target, we suddenly turn off the target. And after like 100 to 150 milliseconds later, we turned on the target at the destination location, and monkey makes saccade. So somehow the attention is modulated during this gap period. Then the monkey's the saccade latency become much shorter compared to the usual visually guided saccade, like this long. But this one, he makes saccade as soon as the target appear. And delayed saccade, we can delay the saccade after the visual stimuli onset. So while monkey is fixating on the target, we show the target, the peripheral or like destination target. But monkey have to stay on the target until this fixation target disappear. And then monkey makes saccade. So the turn off, the disappearing of the fixation target is kind of like go signal. A memory guided saccade, so we flash the destination target briefly during the monkey fixating on the target. And once the fixation target disappears, monkey makes saccade to the remembered target location. There is no visual stimulus, visual target at the onset of saccade and during the saccade. And then we turn on the target once the saccade is landed. And scanning saccade, we show several targets simultaneously, and then monkey choose where to go and when to go. And here is a simple spike activity of the Purkinje cell in the oculomotor vermis for 5 different types of saccade that I just talked. Basically, the firing patterns are all same for all different types of saccade, like targeting, express, scanning, delayed, and memory. As you see, the data is aligned on the saccade onset. Saccade onset is here. And then this is for ipsiversive saccade, and this is for contraversive saccade. And this is one neuron, another neuron, third neuron, like that. And as you see, the firing are the same for all types of saccade, like burst type, burst for all saccade, like burst pods for all saccade. Sometimes different. The scanning saccade showed different firing pattern for this neuron, but like basically all the same. And this is the summary of all like 55 neurons. So this one, the targeting saccade was compared with express saccade, and blue means the express saccade firing was weaker than the targeting saccade, and white means no difference. So this is one neuron, and the second, this is another neuron. So this neuron, the activity was weaker for express saccade, but this neuron, express and targeting saccade are no different. But this one, red means the targeting saccade was stronger than the express saccade. So basically just invert this thing, this blue and this red. So, and just in summary, most of the neurons are no different. Like basically there are large white part here. And some are stronger, some are weaker. So, and then I wondered whether it could be depend on burst type or non-burst type, like neurons firing pattern. So I just split it into like half. Like the activity pattern was mostly burst, and then I just categorized to the burst type, and the others are other types. But as you see, there is no really particular pattern, like black and grays are all there. So in summary, cerebellum fire for all types of saccade and similarly. And this is the adaptation time course for 5 different types of saccade. This is targeting saccade, the visually guided saccade. It's typical nice exponential time course. And express saccade, it also follows the exponential time course, but it is a little more variable, trial to trial. And scanning saccade, this is weird one. So as soon as the adaptation paradigm starts, it suddenly jumps to like low again. And then it's almost just stay there. And delayed saccade, it is similar to the targeting saccade. It follows a nice time exponential time course. That's slightly more variable. And memory guided saccade, this is really fast, much faster than the targeting saccade. So time course are different. And this shows the adaptation transfer. So in this experiment, we adapted targeting saccade and then tested express saccade and other type of saccade. So we only induced targeting saccade. We didn't induce adaptation in the express saccade. So this gray is a targeting saccade adaptation. And during adaptation, we tested the express saccade 3 times. And as you see, as the targeting saccade adapt, the gain decreased. The express saccade gain also decreased. And scanning saccade too. And delayed saccade and memory guided saccade. So targeting saccade adaptation transferred or affected all other types of saccade. And express saccade adaptation, we also tested induced adaptation in all other types of saccade. And express saccade adaptation affected targeting saccade too, but not to the scanning saccade. It didn't change. Not delayed saccade or memory guided saccade. And scanning saccade adaptation, it affected to the targeting saccade and express saccade, but not really on the delayed saccade and memory guided saccade. And delayed saccade adaptation affected all like targeting, express, scanning, and memory. And memory guided saccade adaptation didn't really affect targeting saccade, express saccade, scanning saccade, but it affected delayed saccade. So transfer pattern to the other types of saccade are different in a different saccade types. So this one just summarizes the transfer experiment. Green means transfer, red means no transfer. So basically targeting saccade transferred to all other types of saccade, and memory guided saccade didn't really affect it. So the targeting saccade and the express saccade adaptations are quite different. So I guess the adaptation mechanism of those targeting and memory guided saccade could be different. So I decided to examine further difference of these two types of saccade adaptation. And in 1999, Chris Noto and Rick and Albert found adaptation field in the visually guided saccade, the targeting saccade. So in this experiment, they induced adaptation on the rightward 15-degree saccade. So now after the adaptation, this saccade amplitude was reduced, so it's now like 12 degree. And after the adaptation, they examined the different direction of saccade, like slightly up, further up, slightly down, and further down. And before adaptation, they also, of course, corrected those saccade to those directions. And as you see, the adapted saccade, the amplitude change of the adapted saccade is the largest. And then if you go up, slightly up, the change, the amplitude change is smaller and then smaller. And if you go down, the amplitude change is smaller and smaller. And also the different amplitude, but same direction, also affected. And then they plotted this adaptation field. So here is 15-degree adapted saccade. The gain change was 23%. And slightly downward, this one, this saccade, the gain change was like 18 degree. And further down, this one was only 11 degree. So it transfers less and less as the saccade vector was deviated from the adapted saccade. So I examined this phenomenon in the memory guided saccade. Oh yeah, this is called adaptation field. So this is our data. And visually guided saccade is blue. So as the previous slide showed, the adapted saccade here, 17.5 degree, I adapted, showed the largest gain change. And as the amplitude decrease, the amount of gain change decrease. But the memory guided saccade, it's basically just flat. So there is no like mountain shape adaptation field here. And next one is error signal, spatial memory of error signal. Oops. In this experiment, Rick and Albert tested the different size of the error backstep, the visual error, like what size of the visual error induced the adaptation best. And this shows the y-axis is percent gain change, and x-axis is error size, like 30% backstep, 40% backstep. As you see, like 20 to like 30% backstep induced the largest adaptation. And as the adaptation, as the backstep size increase, the amount of gain change decrease. And here is our data. The visually guided saccade induced the best 35% backstep induced the largest gain change. And as the backstep size increased, like 55% and 75%, the amount of gain change decreased like this. But memory guided saccade, it wasn't really decreased. So the 75% backstep induced adaptation as good as 75%. So here again, the visually guided saccade adaptation and memory guided saccade adaptation are different. And last one is temporal property of error signal. In this experiment, Schaefer and Albert changed the backstep timing, the visual error timing. So they blank the target, turn off the target after the saccade end. And then after a certain period of time, they showed the target at the backstep location. So the usual adaptation, there is no blank, so it's 0. The shorter blanking is the best to induce adaptation. So it is best to show the visual error as soon as possible after the target. That is the best way to induce adaptation. And then as the blank duration increases, that means the error delays, the amount of adaptation decreases. So it doesn't really induce adaptation. Yeah. And then this is our data. Consistent with the previous study, the 0 millisecond delay is the best adaptation for visually guided saccade, blue, and 200 millisecond delays made the adaptation half, and 400 millisecond delay made like quarter. But memory guided saccade, it didn't decrease as much as visually guided saccade. So even 400 millisecond delay, it still induces significant amount of adaptation. But you might wonder, like, memory guided saccade gain could be just changed by blanking without any adaptation. So in this control experiment, I just blanked the target after the saccade, no backstep. So in that experiment, the gain wasn't really changed. So this gain change was induced by adaptation, not just by blanking or fatigue or something other. All right, so again, the error signal, temporal property of error signal are also different in the memory guided saccade and visually guided saccade. So to summarize so far, the visually guided saccade and memory guided saccade, the time course is different, transfer was different, and other properties are different. So that all suggests the brain mechanism for those adaptations could be different. And so here is what I'm thinking. For the visually guided saccade, the visual signal is processed. Visual signal is transformed to the saccade signal and then induce visually guided saccade. So to think about the brain mechanism, let's put like here it's superior colliculus and here cerebellum and brainstem are in the saccade generator. And we know the adaptation site for the visually guided saccade is in the cerebellum. Memory guided saccade, the visual signal is first transformed to the working memory, spatial memory, and then this spatial working memory signal is transformed to the saccade and then memory guided saccade. So visually guided saccade adaptation transferred to the memory guided saccade. So the adaptation site must be somewhere around here because the visually guided saccade adaptation affected memory guided saccade. So the adaptation site have to be somewhere that shares both saccade types, must be on the pathway of both saccade types. On the other hand, the memory guided saccade didn't affect the visually guided saccade. So it should be somewhere independent from the visually guided saccade pathway. So in the first experiment, I recorded superior colliculus to examine whether the adaptation site of memory-guided saccade is upstream or downstream of the superior colliculus. So we know the visually guided saccade adaptation site is the cerebellum. And then we know the colliculus activity doesn't change. So the visually guided saccade adaptation, no change here, but it changed after this. So if the memory-guided adaptation site is downstream of the superior colliculus, like here, the colliculus activity shouldn't change during memory-guided saccade. But if it's upstream of the superior colliculus, like here, then the signal is already changed at the superior colliculus level. Okay. So this is visually guided saccade adaptation data. Y-axis is saccade amplitude, x-axis is trial number, and gain changed by the adaptation. The amplitude changed by the adaptation. So each dot represents each saccade trial. And before the adaptation, I also collected different amplitude of saccade. And the color indicate the firing of superior colliculus during saccade. Yellow means stronger, blue is no activity. And as you see, as the amplitude, even though the amplitude decreased, the firing didn't really change. It's yellow all the way. So after the adaptation, it's already like 15 degrees. It was almost blue before the adaptation, but it's still yellow. So the visually guided saccade adaptation, the colliculus activity for the saccade motor signal didn't change. Okay, so for more quantitative analysis, I plotted like this. So now y-axis is the number of spikes, like this color, it's now y-axis. And x-axis is saccade amplitude here. And this is the pre-adaptation data. So those here's data are plotted here. And saccade amplitude, for example, like 20 degree saccade, the neuron fired 13 spikes here. And for 10 degree saccade, no spikes. 10 degree saccade, no spikes here. And this is called like movement field of this neuron. And then I plotted adaptation data on this movement field. So I selected 50 largest amplitude saccade as no-adapted saccade. And it is here on this plot, like 20 degree, 13 degree, 13 spikes.
Yoshiko.
And I selected 50 smallest amplitude saccade as adapted saccade. And it is here, like the number of the saccade amplitude decreased, but the number of spikes didn't really decrease. And I also selected pre-adaptation saccade, which is the same amplitude of adapted saccade, but before the adaptation. And it is here. Of course, this should be in the movement field. So this red and green are completely different. That's the superior colliculus activity during visually guided saccade adaptation was not changed. So that suggests downstream, the adaptation site is downstream of the superior colliculus, which is cerebellum. So that makes sense. And this is memory guided saccade adaptation. So as you see, these and these are really different. Like blue is blue after the adaptation, green is green before and after the adaptation, yellow is yellow before and after the adaptation. So on this movement field plot, so this is this neuron's movement field. No adapted, adapted, and pre-adaptation, it's all in the movement field. So that suggests, that indicate the colliculus activity during memory-guided adaptation decreased during the adaptation. So it changed. That suggests the adaptation site for the memory-guided saccade is upstream of the superior colliculus. Okay, so now we know the adaptation site is upstream of the superior colliculus. And the next question is how those two adaptation site switches. Like one possibility is when we adapt visually guided saccade, it only derives this cerebral adaptation site. When we adapt memory guided saccade, it's only derive the memory guided adaptation site. Another possibility is it kind of gradually change. Sometimes we use both, sometimes only one side, sometimes only one side. So to test, to address which is true, I examined short memory guided saccade. So short memory guided saccade is like this. So memory guided saccade, target flash, and monkey wait until the fixation target disappear and then make saccade. So that means monkey has to hold the memories, the spatial memory during this waiting time. So the longer this waiting time, the monkey has to use more spatial memory holding function. The shorter, it's less. And for visually guided saccade, 0 millisecond wait, so no spatial memory need to be worked. So for the long memory guided saccade that I showed so far, this pink one, I used 1 and 1.2 seconds for this wait duration. But short memory guided saccade, I made it half, 0.5 and 0.6 seconds. So if it is this case, then short memory guided saccade adaptation and memory guided saccade adaptation should be the same, but different from visually guided saccade. But if it is this case, then short memory guided adaptation characteristic should be in between of this visually guided and long memory guided saccade. So this is the result. This one is the adaptation field that I showed earlier. The visually guided showed a nice mountain shape, and long memory guided saccade was basically flat, and short memory guided saccade, it was in between of those two. And this one is a backstep size, like 35 seconds induced the best and 75% the too large. So 75% backstep is too large to induce visually guided saccade, but it was fine for memory guided saccade. And short memory guided saccade was again in between those two. And this is a delay of the backstep. So 400 milliseconds didn't induce good adaptation in the visually guided saccade, but it was okay for the memory guided saccade. And short memory guided saccade adaptation was middle of them. And this one, I tested visually guided saccade after short memory guided saccade adaptation and long memory guided saccade adaptation. So checking the transfer. So after memory guided saccade adaptation, there was no transfer to the visually guided saccade. So pure visually guided saccade and memory guided saccade adaptation are independent, which is consistent with that I showed earlier. But short memory guided saccade adaptation, it affects about 30% to the visually guided saccade. So short memory guided saccade adaptation affected the visually guided saccade. And this is the colliculus activity for short memory guided saccade adaptation. As you see, it decreased like the visually guided saccade, but it almost on the movement field like this. So it's again, short memory guided saccade was kind of middle of this and this. So all of those data suggest it uses this type, not like just completely switching both adaptation site. Okay, so the remaining question is where is this adaptation site for the memory guided saccade? I don't know yet, but just speculating. So it should be upstream of the superior colliculus. And there are frontal eye field and basal ganglia upstream of the superior colliculus. And further upstream, there are DLP and LIP. So to speculate like where should I go to the next? I analyzed the colliculus activity like this. So this is the visual motor neuron in the superior colliculus during memory guided saccade. So target flash here and monkey make saccade here. So I split to 4 intervals, like visual activity, like target flash here. So this is visual activity. And this is memory hold interval. So I split into 2, like early working memory activity, early working memory interval, just half and half. Late working memory interval, and saccade related interval. So this neuron, visual motor neuron, first the neuron fired, responded to this visual input, and then sustained the activity during whole memory waiting time and burst for saccade. And this blue was no adapted saccade, and this red is adapted saccade. So the visual activity was not changed. And early working memory interval, it's not really different, blue and red, but slightly different in the later part. But in the late working memory, red is smaller than the blue. And during saccade, it of course different as I showed earlier. So on this movement field or visual field plot, the visual activity, red and blue, no change. Early working memory, it's still no change. So late working memory, it's slightly decreased. And saccade related activity, it completely decreased. So the change seems started already in the late working memory period preceding to the saccade related activity. So to think about the brain structure, so the signal, first it visual signal, and then during this interval, the visual signal is transformed to the working memory, and the later part, working memory is transformed to saccade, and then saccade. So the adaptation could be already happening during this transformation period. So based on that, I guess here should be good than further upstream, because the visual signal is first transformed to the higher upstream, visual to working memory transformation, and then working memory to saccade transformation here, and then saccade at superior colliculus, and further down brainstem and cerebellum. Indeed, the Mickey Goldberg group examined LIP activity during memory-guided saccade, and they showed no change during the adaptation. So that suggests the adaptation site is downstream of the LIP. So this is consistent with their finding. Okay, and how could the adaptation is induced? We know the superior colliculus, for the visually guided saccade adaptation, the superior colliculus send the error signal to the cerebellum through the inferior olive. So I'm wondering if this signal is also used to induce adaptation in those areas. I don't know yet. And why? There are 2 sites to induce adaptation. Just cerebellum can induce all adaptation, but somehow there are 2 sites of adaptation, different adaptation sites. We don't know, but it could be interesting to study. Okay, that's it. Thank you very much.
Thank you, Yoshiko. Maybe I can start the questions. Yoshiko, is it possible that the cerebellum
Battery full.
Sorry, my speaker turned off.
is the cerebellum also maybe it's possible that the cerebellum is also involved in the memory guided saccades, but a different part of the cerebellum, possibly the lateral cerebellum. What do you think about the role of the cerebellum in adaptation of memory guided saccades?
So for this, like when the weight duration is short, then cerebellum is also involved. But yeah, different, like lateral cerebellum, it also like have function for timing, and memory guided saccade have timing, like have to wait until the target disappear and then make saccade. So timing could be also involved. So yeah, lateral cerebellum could be involved too.
Can I follow up on that question?
Yes, please.
Of course, Sasha, go ahead.
Great. Hi, Yoshiko. That was beautiful.
Thank
So,
you. Nice to see you.
So I've just been analyzing Hirofumi Fujita's anatomical data. It's in mouse, but we are so struck by the density of projections from the dentate to the superior colliculus relative to the fastigial. It is unbelievably strong. I mean, it's really strong, and different parts of the dentate. And so sort of thinking, I think I saw Shogo on the call, and so maybe he could comment, but Mike Mock in the eye blink conditioning literature showed really nicely that when there's a delay, there's like, it's called trace conditioning, stupid names, but when there's a long delay between the tone and the puff or whatever, then that's a prefrontal hippocampal circuit that maintains that delay. But right at the end, the information's given to the cerebellum. So one possibility I'd like to suggest is that something like the cerebellum really is upstream of the superior colliculus in both cases, but maybe different collicular neurons. And as Reza was suggesting, maybe the lateral dentate, which is getting a lot of lovely input from the cerebral cortex via both the inferior olive mesodiencephalic junction and mossy fibers. So maybe the cerebellum is really upstream of everything, but just to emphasize Reza's point, it's different parts.
I see. Yeah, like when I read like Dr. Tanaka's paper for timing, like my mind, like all saccade system, cerebellum is downstream of like thalamus, basal ganglia, and like of course cortex. But for the timing study, timing field of the cerebellum is upstream of the basal ganglia, which is kind of eye-opening, right? It could be different in the different system. So yeah, as you said, the memory guided saccade is way more complicated than the visually guided saccade, and it involves timing and have to wait, and involves spatial memory. So yeah, like the cerebellum could be upstream of the basal ganglia and like those areas. So
yeah, just to emphasize what you're just saying, in the mouse, which really is fundamentally similar to the primate, just less of the interesting stuff. In the mouse, the ventral dentate, just like it does in the primate, projects to the thalamic nuclei like parafascicularis and centrolateral, which are presynaptic to the basal ganglia. So there's lots of ways for the cerebellum to tell the basal ganglia and the cortex what to tell the colliculus.
Yeah.
Anyhow, beautiful work.
Thank you.
Rich, you had a question.
Yeah, that was really a lovely talk.
Thank you very much.
Both the behavior and the neurophysiology. So I'm really the behavior's got these interesting signatures, and in the reaching world, in some ways, people have kind of made a distinction in the human reaching world between adaptation and kind of changes in the execution system as adaptation, and changes in kind of a selection as another way of adjusting, right? So, kind of more of a strategic process, and kind of made a distinction between cerebellar-based and non-cerebellar-based. And a couple of your behavioral signatures of the memory-guided are very much akin to what you see when you see strategy-based changes. So, for instance, the broad generalization that you observe for your memory-guided saccades and the resistance to the temporal delay. These are the kinds of things you see when people adapt by kind of strategically choosing to re-aim to a different location. And that maybe also in some sense has some similar feel to your shift from a cerebellar to a upstream or non-cerebellar locus. So I wanted to hear your thoughts on that. And I'm trying to think, though, places where the things don't really align might be on the washout part, because with kind of more strategic changes in the human literature, you get very fast washout. You don't have to see a long washout period. And I don't know if I don't recall whether there was a real difference in the washout for your memory guided task versus your visually guided task.
Yeah, I have, let me. Maybe I stop sharing first and then. Something not right, sorry. So, you see my screen?
No, we
Yeah.
don't see it.
Okay, so washout. So targeting saccade, like after the adaptation, we suddenly stop the adaptation paradigm. It kind of jump up.
Yoshiko, we can't see your screen.
No, you cannot see.
There we go.
There it is.
Okay, so targeting saccade, it's kind of like decrease, and then once we stop the adaptation paradigm, it's jump back here. So if like retention or washout, like if it's strategic, it's more so like switching, like adapt and jump back and adapt and jump back. So for targeting saccade, it's kind of both, like jump back and recover. So this slow recovery is really like adapted, not strategic. In this
Yeah, that certainly looks.
strategic. So if you see memory guided saccade, the jump is definitely larger than the targeting saccade. So yeah, I agree. There are more like strategic component in this memory guided saccade adaptation.
I see, yeah. So there really is actually, and many of the behavioral signatures really line up well with what people have described between kind of adaptation and, like strategic re-aiming or whatever.
Yeah.
I see.
And the scanning saccade is really weird. It's just jumps, so really strategic, but it stays there when we stop. So this is most tricky one.
I had a second question, if I.
Yes, please.
So I was also struck by what seems like the opposite effects of memory-guided and express saccades, and that the express saccades attenuated the rate of learning, whereas the memory-guided accelerated. And I'm thinking about, one case, right? In the express saccade, you lose your start position, right? That goes blank. Whereas in the memory guided, you lose your target position. And I didn't know whether you thought that somehow having where you're starting from versus where you're going from, that has some contribution to this change in the rate of adaptation.
It involves more attention, higher order. Yeah.
I mean, this is sort of before I was thinking about that the memory guided maybe is more about kind of a selection level type change, but any thoughts about, so why in the express saccade condition do you think you get this attenuation in the rate of adaptation?
Sorry, attenuation.
In the express saccade case, right, if I understood correctly, the rate of adaptation was slower.
Yeah, than memory guided saccade.
No, compared to the compared to
Oh.
the basic and compared to the memory guided.
Okay.
And I was wondering what you thought leads to the slower rate of adaptation in the express condition.
Yeah, so express saccade, so the gap paradigm doesn't induce express saccade 100% of the time. Like, it, if we plot the number of saccade and the saccade reaction time, it gives bimodal distribution. So it's about like in the whole trial, only like, let's say like less than half of the saccade was express saccade and the other was like regular latency saccade, even though we do gap paradigm. So when monkey made regular latency saccade, we just blanked the target. We backstepped the target only when monkey made express saccade. So in the whole trial, only about less than half of the time we gave the backstep. So that might affect the time course of the express saccade.
Okay.
Although this is only plotted express saccade, but yeah.
But if you were betting on things, you'd be putting the express saccade in the same neural regime as visually guided. Okay.
Yeah, express and targeting saccade transferred each other, and that seems similar in both.
Well, thank you very much.
Yeah, thank you.
Thank you, Rich. Shogo, you have a question.
Yes, thank you. Thank you very much for your really wonderful presentation. It's really beautiful. My question is about the complex spike during the memory guided saccade. And so if the complex spike happens in the memory guided saccade, then the interesting gap between the complex spike and the cerebral learning, because your data suggests the cerebral learning didn't happen in the memory guided saccade task adaptation. So could you give me your the observation for the complex spike in the memory guided saccade, and also you give me your idea for the potential gap.
Yeah, that is really interesting. I mean, yeah, I should have done. But for this simple spike, when I recorded simple spike, I didn't induce adaptation. I didn't do backstep. I just tested all different types of saccade. So I don't know whether the complex spike fire, but I guess like it should fire. Complex spike is basically just visual.
Yes, I expect so.
Yeah, I would be surprised if it didn't. There's no complex spike for the memory guided saccade, but then it should induce adaptation. So yeah, it's.
Yeah, then in the memory guided saccade case, somehow the complex spike driven learning is suppressed, right? That also can be very interesting.
Yeah, right. Yeah, thank you.
Okay, thank you so much.
Solomon had a question.
Yeah, yeah, really fantastic. I guess it's maybe following the question, maybe a normative question. Is there any kind of logic you can say why in visually guided saccade you cannot have that type of adaptation that you see in the memory guided?
Why? So like.
Why would you use this kind of? I'm sorry.
More strategic learning.
Yeah, exactly.
I think so. Could be we human might use more strategic, because compared to monkey and human experiment, the adaptation in human is much faster in the visually guided saccade compared to monkey. Like we can adapt completely, like at most 100 trials, but monkey takes more than 1,000 trials. So, but monkey probably not really just doing. They don't use strategy. I don't know. So humans' adaptation might be similar to the memory guided saccade. I don't think anybody has done in like, backstep timing has been done in human, but I'm not sure whether the movement field and backstep, no, backstep size has been done in human, like Albert did with Joe, but I don't think human, I cannot
I see.
remember whether
So-
backstep timing.
Okay, so just a quick follow. So that would imply that in the monkeys, memory guided would be much slower than targeting. Is that true?
Memory guided is faster than
It is
targeting.
faster.
Yeah. And human is faster than monkey. So monkey's
Okay.
memory could similar to human visual.
Okay, thanks.
Yoshiko, would you mind if I ask you a question?
Yeah, of course, please.
Beautiful talk. I was interested in asking about your colliculus data. I know Ziad Hafid had this paper that described that there are collicular neurons that respond differently to the same amplitude memory guided versus visual saccades. I was wondering first if you see this pattern for all the collicular neurons that you have, and if you see any difference between the the different types of saccades that you see in the colliculus.
Yeah, so there are many different type of colliculus neuron, like visual neuron, just briefly view, visual motor and motor. So visually guided saccade, the visual response to target is really close. So it's kind of both signals are overlap, but memory guided saccade, the visual response and saccade are like really separated. So you can purely analyze, can dissociate visual and saccade signal. So probably that is one reason the firing is different in visually guided and memory guided saccade.
I think the ones that they used were only motor. So at least their plots seem to be well separated. There was no visual component in that.
No visual component override to the saccade?
Yeah, in the example that at least I saw in their paper. It was probably from the deeper layers. That's
Yeah.
the motor component.
So sorry, what was your question?
So what I'm asking is that, do you see a difference in the response of the neurons between the visually targets, the targeting saccades and memory guidance, if they have the same amplitude? And also, are they different in their response to the adaptation that is happening?
So for the adaptation, I can only record one neuron at a time. So I cannot directly compare. Like I cannot induce both adaptation and check. So I cannot answer that question. But the first one, so it's really different for the visually guided saccade and memory guided saccade. Because as I said, like probably there are some neurons had really short visual response and short saccade burst, so it could be dissociated. But the duration of the burst is really different neuron to neuron. So most neuron that I record, it's like override both when I record visually guided saccade. So if I compare like memory guide and visual guide, it's gonna be different in most of the neuron because of that.
And all of them showed the same pattern, same beautiful pattern that you showed, that for the visual guided, the response did not change. For the memory guided, it was changing.
Not all, no.
Oh, not all.
Yeah, not all. Like this is physiology. There are many variables. Like the neuron which doesn't change at all, the neuron that changes. Yeah. But that is the best typical neuron. But most of,
do you have a percentage of how many showed this pattern and how many did not show any pattern?
Oh yeah, I do.
Adaptation.
I do. Yeah. Let me memory guide. Yes. So this one shows like how many units. Visually guided saccade adaptation. So this black bar means no difference. Or like, I mean, this adapted saccade and before adaptation was significantly different. That means adapted saccade was outside of the movement field. So basically, visually guided saccade, all were different. That means visually guided saccade was basically no change. It's out of the movement field. And short memory guided saccade, it's like 25% was similar to the long memory guided saccade. Like it's just changed. And like 75% was changed. And memory guided saccade, like about 75% of the neuron was not changed. And like 25% changed. And this is gain increase adaptation. I only showed gain decrease today, but gain increase adaptation somehow all neurons showed no change.
Almost all your neurons, I think, showed the pattern.
Yeah. And if I check like more long wait duration memory guided saccade, it may change this percentage.
And are most of these visual motor neurons?
Yeah, visual motor. Yeah.
Thank you so much.
Yeah, thank you.
All right, Rich, you have another question?
If no one else does, yeah, so work really also though really kind of changes sort of our basic picture in the sense that we usually think that you're reaching for a target, but the kind of the sensory prediction comes from the movement itself, right? From kind of an efference copy from the movement.
Yeah.
But here that would really, you'd expect then to see, you know, if that was sort of the way things worked, you'd expect to see kind of cerebellar-based adaptation in the memory-guided situation. And you know, obviously, you know, your results say that things really change, that we shouldn't just think of the target as kind of a proxy for where you expect your, you know, effector your eyes or your hand to get to, but it's really quite a different story. It seems that, you know, that's one message I take from your talk here.
Yeah, thank you.
Yoshiko, thank you so much. It's been great to have you, and thank you for all the questions, everyone. Hope you have a good Tuesday.
Yeah, thank you very much.
Fantastic, Yoshiko.
Bye.
Take
Thank
care.
you.
Thank you.